We're starting chapter 19 on the blood, a mobile connective tissue, which is a slurry in its nature. It basically provides the transportation for all elements required for life that have to be distributed around the body. This is a consequence of piling cells up. You can put cells together as densely as you want, but you have to get all the living conditions delivered. That includes gases from the environment, nutrients in solid and liquid form, wastes have to be removed, hormones have to be delivered, regulatory elements in the blood, proteins circulate in the blood, maintaining the osmotic balance, etc., etc. That means that a lot of conditions are regulated by the blood in its presence. pH with the bicarbonate system is regulated within a relatively narrow range. Plasma blood set 0.7, 0.4, slightly basic, but the range is normally only between 735 and 745, so fairly tightly controlled. Because of the temperature, the latent heat of water, water can hold and transport heat. So thermoregulation and body temperature is managed on a whole body basis by the circulating blood and fluids. Blood also has the means of plugging up holes if they're small enough. Fluid loss and clotting are something that we will attribute to the blood. Finally, in chapter 22 is the critical examination of this. We have the ability to protect the body through antibodies and white blood cells. And this kind of chemical defense system is our way of managing the arms race. Right now, COVID-19 is on everyone's mind, and it is a question of COVID-19. How do we develop effective antibodies? How do we make effective vaccines? Those are still open questions in our confrontation of this new variety of pathogen. Here's a typical blood smear. Now, what you do is you take whole blood. Now, you typically fix it and stain it. That's why the picture looks so pretty. But what we have here is the presence of three different types of cells. These are classes of cells, and the first and most apparent are these pink, clear, disc-shaped red blood cells, by far the most numerous in this picture. Here is one red blood cell that's lying flat on the slide, and you can see it's got a darker rim and a kind of a hole in the center. It's because this is sort of a disc that's called a biconcave disc. There's actually a curve in the center from each side that almost meets, increasing the surface area of this red blood cell. Primary job of the red blood cell, carry oxygen. These larger cells, notice also something about the red blood cell, they appear clearer. It's because they don't have a nucleus. As they are formed, the nucleus is thrown out. With most of the other organelles, we're going to just pack this with a special oxygen-carrying molecule called hemoglobin. And hemoglobin is what will be the primary oxygen transport mechanism. Now, it has some secondary chemical effects. It does interact with CO2 to some effect. It does affect the pH in some ways. But since 99% of the oxygen that's carried is carried by hemoglobin, you know its primary effect is pretty fixed. Now, these four larger cells have odd-shaped, dark purple nuclei. These are very convoluted, and they regularly appear in this condition. This cell over here, you can kind of see a lighter rim across the bottom. That's the whole cytoplasm. That's a white blood cell called a monocyte. And white blood cells retain all the organelles and full cellular capacities. White blood cells are there for protection. These dots, these purple dots, are the platelets. The platelets are there for blood clotting and developed by fragmentation from a large progenitor cell. Let's take a look at the basic components of the blood, starting here with red blood cells. They're very numerous. Every milliliter, cubic millimeter, contains 4 to 6 million of them. They are relatively tiny. Their job, transport oxygen. They help somewhat in CO2. And their source, as in all blood cells, is the red bone marrow. Now, the division between the fluid, the formed element of the blood, by the way, sometimes you'll see blood cells referred to as sediment because they literally are. They're not dissolved. When you spin in a centrifuge, you get this distribution. The formed elements, about 45% of the volume, you can see on the bottom. By the way, do you see that light kind of ivory colored band? That's because the red blood cells fall fastest. And in the final sedimentation, the white blood cells form this buffy layer on top of it. 45% will be blood cells. 55% will be this kind of yellow colored solution called plasma. This proportion, the proportion of blood cells in whole blood, is called the hematocrit. Hematocrit. And you can see hematocrits changing as a result of disease. If you have a disease that lyses your red blood cell and the hematocrit goes down, that's one of the first signs you have a blood problem. If you move to higher altitudes and constantly are breathing under an atmosphere that is lower in oxygen content, your hematocrit can increase. Hemorrhage would cause, so let's say you have a hemorrhage that's controlled and you drink a lot of fluid, your hematocrit would be lowered and erythropoietin would increase and bring it back up to normal levels and so forth. So let's just for a moment take a look at these formed elements. The red blood cells are only of one type. They're very small. White blood cells are of many types. These three are called granulocytes because they have these granules filling the cytoplasm. We're going to go through them one at a time. Their whole idea is to fight infection, and red bone marrow is their site of production. We can see here basophils, eosinophils, and neutrophils are the three kinds of granulocytes. There are then agranular leukocytes, leukocytes meaning white blood cells. Lymphocytes and monocytes are those cells that are agranular. These are also, the lymphocytes are named because these are responsible for specific immunity. The granulocytes are responsible for a general immunity that we all have. Finally, this platelet or thrombocytes, thrombosis is clotting. A thrombus is a clot, so these are the clotting cells. They are relatively small because they come from a large cell that breaks apart. So these are basically just fragments. And the idea, they're produced in the red bone marrow, but the idea is they're going to clot the blood. All of these develop in red bone marrow. So there's a rich blood supply nourishing the living cells of the red bone marrow. And they have the appearance of a typical cell. Hemocytoblast, blood cell to become, are the progenitor cells that then differentiate down different pathways under the influence of chemicals called colony stimulating factors. So we see a main division producing this myeloid stem cell, and then multi-CSF acts on this to produce these three different tracts and their subsets down here. Over here, EPO, erythropoietin, we have mentioned. Erythropoietin causes this myeloid stem cell to develop through stages, actually shrinking the cell. Look at this ejection of the nucleus and many of the organelles, finally forming the mature erythrocyte or red blood cell. Over here we have another myeloid stem cell going through a course of development to form this odd cell, a very large cell you see in the bone marrow, huge nucleus, and protrusions or little structures sticking out of its cell membrane. This is called a megakaryocyte. Once you've seen one, you never forget it. It matures in the red bone marrow on a constant basis and basically then just explodes. Each one of these fragments, called a platelet, has a tiny bit of cytoplasm, a membrane, and a small fragment of the nucleus. This makes it a competent platelet. Over here, the myeloid stem cell, the general myeloid colony-stimulating factor, produces several stages of development, and we do recognize a difference here between a myeloblast and a monoblast. These are different developmental pathways. The myeloblast gives rise to the granular lymphocytes, basophils, eosinophils, and neutrophils. And the monoblast stimulating factor gives rise to monocyte. And we have to go all the way up here to the hemocytoblast to find the track that leads to the lymphoid stem cell, developing down here to the lymphocyte. These are the specific immunity cells. When we look at the other side of the blood, not the formed elements, but the plasma, we find a very carefully constructed material, not just for its dissolved materials, but for its osmotic balance and pH. So 92% water. This is our blood volume. This is our osmotic pressure. This is our transport medium. And almost all of the water is absorbed from our intestine and our diet. We drink to hydrate. Now, I do want to point out there's a thing called metabolic water. There are some reactions, and notably those that are involved in producing ATP, that generate water as a product. That I'd call metabolic water. It's about 300 mils a day. So this is not totally true. Within, we see some major chemical components in the plasma. Plasma proteins make up 7 to 8% of the weight. Albumin, globulins, and fibrinogens are classes of molecules produced in the liver and basically used to maintain this necessary concentration of proteins in the blood. Salts come from, those are the dissolved ions. They are there for osmotic pressure, for pH, and as metabolic cofactors. They're absorbed in diet. Gases come from the respiratory tract. Oxygen and carbon dioxide are our main two. And cellular respiration is the end product, the cellular activity that results when you have a respiratory system. When you begin with breathing that gas in, you have to ask, what's the oxygen used for? Well, cellular respiration. Lungs and tissues are the source. Lungs are the source of oxygen. That's the entry point. And tissues back to the lungs are the pathway for CO2. Nutrients, this is from our digestive tract, absorbed largely through the intestines, with our main nutrients being lipids in the form of fats, oils, waxes, glucose as an example of carbohydrate, and very diverse in diet, and amino acids and proteins. These are our food stuffs that have to be broken down from food to nutrients and absorbed by the intestine. If you break down this one, protein, you generate ammonia. The body processes that by making urea and uric acid, which is filtered out and formed into urine and basically excreted by the kidneys, although urea is produced by the liver. So the tissues produce ammonia, which forms ammonium. The blood going through the liver is converted to urea by the urea cycle, and that urea is filtered out by the kidneys. Kind of a complicated process, but since ammonia is such deadly poison, it's the only way we can have a multicellular body that feeds on protein. Others. This could be a very long list because we have so many things that act in low concentration, like hormones and vitamins, but also many traces. They aid metabolism, they're essential, and their sources are varied in the body. Let's start with plasma protein. Albumin is the most abundant, and its main job, osmotic balance. It also is a carrier, a protein carrier that can transport fatty acids and hormones. Now, remember that fatty acids are lipids, and there are steroid hormones, there are lipid hormones. So we would expect those two things to have problems with solubility. So having a carrier that can bind them and basically move through the water solution easily kind of makes a lot of sense. When we talk about plasma proteins, globulins are the next. Globulins were named for their shape, kind of globe shape. And these are the antibodies that our body manufactures through the immune system. Globulins can also act as carriers for hormones. Finally, fibrinogen is a plasma protein. It forms fibrin during blood clotting, so the soluble form is fibrinogen. And when we have a damaged area and damaged chemicals present, it is converted into fibrin. Plasma without fibrinogen is called serum. So here we have that a basic plasma sheet with everything that we have previewed in the plasma proteins. We've already talked about salts, gases, nutrients, nitrogenous wastes, and other as a prelude to looking at the blood carefully. The first item of study, kind of hard to decide what is the most important molecule for the human body. Well, we have to set up a criterion first, and let's pick the one that you die quickest without, and that's oxygen. It's also why we study this molecule first. It's an element of the blood. It is a protein that is a tetramer. You will notice 1, 2, 3, 4 protein chains, each wrapped around this central heme group. The red globe inside it is an iron. These are 4 different polypeptide chains, but only 2 types. There are 2 alpha chains, shown plain, and 2 beta chains that are shown, 2 of these are alpha chains and 2 of these are beta chains. They've simply drawn in some of the detail on one of the chains. This forms what's called hemoglobin. The heme group represents a binding site that can bind 1 oxygen molecule. So over here, the red blood cell is packed with hemoglobin molecules. Each hemoglobin molecule can bind 1, 2, 3, 4 oxygens for transport to the tissues. 2 alpha and 2 beta chains means that there are 2 genes, an alpha gene and a beta gene. That means different nucleotide sequence, different proteins are produced to produce normal adult hemoglobin. Now, the reason I'm saying that, normal adult hemoglobin, is that that's what we call the 2 alpha, 2 beta, recognizing something important. The hemoglobin in a fetus has 2 alpha and 2 gamma for reasons we'll explain in a moment. There are also disease conditions that change these genes, alpha or beta, and generate a new and usually subvital protein chain. There is, for example, what we call the sickle cell beta chain. And alpha alpha beta beta, alpha alpha beta normal, and alpha alpha normal normal are possible in a person who is heterozygous for this condition. We'll look at that too. Oxygen circulation. We have the depleted molecule here, colored purple, 2 alphas, 2 betas, tinted differently, and 4 heme groups. And colored purple because actually in this state, hemoglobin is deoxygenated, and it has a sort of a bluish, purplish tinge. It's why the veins in the back of your hand have that blue tinge. They're coming back from the tissue in depleted form. But loading oxygen in the lungs, 1, 2, 3, 4, we get oxyhemoglobin, and it definitely changes the color of the blood to a bright scarlet red. The dynamics of any molecule, its kinetics of reaction are important, and we're used to thinking of reactions in the chem lab as producing a yield. It's not unusual to do a reaction under ideal circumstances and get 80% yield as the natural reactivity. But this tells you something important. This is a molecule that is ideal for oxygen transport. 99% of the oxygen is carried in the red blood cells. So this is a specific carrier. It kind of points to the importance of oxygen transport, since oxygen is the limiting molecule in most circumstances of exertion. Better oxygen transport means a better body. It works better, stronger, faster, harder. Gas transport is shown. Hemoglobin is going to load oxygen at high oxygen concentration, but it's going to unload it at low pH. These are also elements of the kinetics of the molecule. So where you run into acid, oxygen's going to dump. Where you run into oxygen, oxygen's going to be absorbed. Now, if you think about the body, the lungs, we're constantly inflating the lungs with oxygen, 20% oxygen in the air. So high concentration in the gas pockets. Run the blood through a capillary in the lungs, and there's high concentration right next door. Across the capillary wall and the alveolus wall, which are very thin, is high oxygen. So if that hemoglobin is unloaded, it's going to basically be bound very tightly to that hemoglobin. If oxygen's going to bind it very tightly until it's full. Now the blood, which is flowing, pushes that loaded cell out and replaces it with a new cell that is unloaded. So there's a constant high concentration gas pocket right next to a low concentration capillary, and diffusion will drive the oxygen across. Now the blood coming in, not just the red blood cells, but the plasma and other carriers have high concentration of CO2, and the gas pocket is very low in CO2. So diffusion is going to drive CO2 out at the lungs. Now, we're not going to deal with this just yet, but we're going to recognize this when that enriched blood, high oxygen, low CO2, leaves the lungs and goes to the tissues. It gets to a capillary bed, let's say somewhere out in the biceps brachii, my favorite muscle. Now, as it gets out into the capillary, molecular exchange is possible. And oxygen in the surrounding tissues has been consumed. It's been consumed by aerobic respiration. Glucose plus oxygen produces CO2 plus water and ATP. So high concentration of oxygen in the blood and on the hemoglobin produces high concentration in the blood and low concentration in the tissues. And in the tissues, That CO2 that's produced produces that higher acid because of this reaction. Here's CO2 and water being produced in tissues. Carbonic acid dissociates into hydrogen ion and bicarbonate. So the tissues that surround the capillaries are low in pH. So the chemical conditions for diffusion and for pH favor the absorption of oxygen in the lungs and the dumping of oxygen in the tissues. We're going to see the reciprocal effect for CO2, the dumping of CO2 in the lungs, and the absorption of CO2 in the blood in the tissues. Hemoglobin does bind some CO2, very, very little. It's called carbaminohemoglobin, and it's kind of a result of an accident, whereas the hemoglobin has 4 chains, and 1, 2, 3, 4 oxygen are absorbed at a specific active site, the heme. Over here, at a junction between the 2 alpha and 2 beta chains, there's one site that can kind of weakly bind a CO2. So maybe 20 to 25% of the CO2 is going to be on the hemoglobin. We'll deal with that and tell you where the other 75% is in just a bit. CO2 is carried more often as a result of this equilibrium. This produces a buffer called bicarbonate, which can absorb excess hydrogen ion if it encounters it. Hydrogen ion is going to be carried in various forms in solution. Hydrogen ion does bind on hemoglobin. Hemoglobin, especially hemoglobin that is deoxygenated, carries a certain amount of hydrogen ion. But the thing I want to reinforce here, because it's going to come up again and again, here and in the respiratory system especially, producing CO2 in tissues which have a water solvent. It's going to produce carbonic acid and hydrogen ion and bicarbonate. So always CO2 production means more hydrogen ion and lowered pH. Other gas transport issues for our system. It turns out that hemoglobin actually binds carbon monoxide more strongly than carbon dioxide or oxygen. What does that mean? That means if you're with a gas, it might have 20% oxygen, but if it also has 20 or 30% carbon monoxide, the carbon monoxide is going to bind the hemoglobin, so that in the presence of enough oxygen in normal air to support life, you might asphyxiate in the presence of carbon monoxide. Carbon monoxide is a gas of choice for asphyxiation. Fetal hemoglobin is a distinct hemoglobin, as I mentioned, 2 alpha and 2 gamma chains, which binds the oxygen more strongly than maternal hemoglobin. Let's think about this, because we just talked about fetal development. Here's the mother side with 2 alpha chains, 2 beta chains, providing an oxygen supply. And here's the placenta side, a mother tissue, but baby side in terms of circulation of blood, circulating the blood through here. Well, if you dump oxygen here, and you have blood of the same type on both sides, they attract the oxygen to the same amount. But if you have fetal hemoglobin and it binds oxygen more strongly, then the oxygen moves from the mother side to the baby side and is dependably bound and carried away. Now, this is because we have, as we called it, an exchange zone between the uterus and the placenta. Excess of red blood cells, called polycythemia, can occur. Basically makes the blood kind of thick. Anemia results when oxygen delivery is insufficient. I want you to look right here at this definition and try to secure it in your mind. This is one of the most confusing points, because people have been taught about iron deficiency anemia or hemolytic anemia. The anemia is not a specific cause, it is a symptom. If your tissue's getting insufficient oxygen, there's a reason. And you look for the specific reason. It is an important disease since we're talking about oxygen delivery. Oxygen delivery means cells at risk. So if it becomes pronounced, cells begin to die. What are the things that can actually cause that? Remembering that oxygen delivery is the issue, you can actually get a reduced oxygen carrying capacity if you lose red blood cells, if you have abnormal hemoglobin. But anemia is not a disease, it's a symptom. The symptoms include, from the outside, you feel tired with low oxygen. Often your skin will get pale. You'll feel cold. Shortness of breath accompanies it because we're talking about hypoxia. Let's look at some of these reasons. Hemorrhagic anemia can occur from a hemorrhage event. Of course, if it's outside and catastrophic, you lose a couple liters of blood and survive. We basically replace that and build it back up. More dangerous is where you have chronic slow bleeding. So you have an aneurysm that ruptures in your intestine, leaking out 10 mils of blood every 15 minutes. It's something you may not feel for several days. There may not be any pain associated with it. That chronic slow bleeding might first be detected as anemia. Hemolytic anemia is something that destroys red blood cells. And so infections do that. Genetics can do that. Malformed or subvital red blood cells. Interestingly enough, there are some drugs administered for one reason that have a side effect of destroying red blood cells. Aplastic anemia occurs when you have, in some ways, compromised the bone marrow. Bone marrow. This is an important element in radiation therapy because bone marrow, and now we're doing so many radiation therapies for cancer, and the radiation is supposed to kill the rapidly dividing cancer cells. Well, it turns out we're turning over our blood cells very quickly, so bone marrow is very active. Radiation has a differential effect on bone marrow and kills those cells. DDT is a toxin that has a specific effect on blood cell production. So again, some drugs like chloramphenicol will affect the blood cell producing cells in the marrow. Nutrition does affect. There used to be a tonic called Geritol that used to advertise for iron poor blood. If you don't have enough iron in your diet, or for some reason its absorption and utilization is compromised, nutritional deficiencies can result. It can also result from hemorrhagic anemia. We're releasing the hemoglobin and the iron into the blood and losing it through excretion. Pernicious anemia results when we don't have enough vitamin B12. So you can see there are many types. Abnormal hemoglobin. If a baby is born normally, the fetal beta chain, the gamma chain, ceases production, and the beta chain is produced. So the blood normally goes from alpha, alpha, gamma, gamma to alpha, alpha, beta, beta. If that does not occur, it produces a condition called thalassemia. That results from abnormal hemoglobin. Our second example is sickle cell disease. Sickle cell disease is caused by a mutation of the beta chain. It is only one nucleotide different, but it produces a beta S protein. And we will look at the causes of that in detail. Here is a blood smear. Now this has been color enhanced, and the background has been colored as well to kind of show you that a person who carries a heterozygous genotype for sickle cell disease can survive And as you can see, some of these blood cells actually look normal. In fact, this figure really shows the biconcave, the depression on the big flat side that's present in normal red blood cells. But if you're normal, you would have beta beta as your genotype. However, if you're heterozygous, beta sickle, then you can assemble hemoglobin that's alpha alpha beta beta, alpha alpha beta sickle, and alpha alpha sickle sickle as hemoglobin molecules. The number of molecules that go into the red blood cell causes the packing to be different. So you can see there's plenty of red blood cells here that actually look pretty normal. But then there's these purple colored ones that have odd shapes. Look at this one, looks like a kind of a handle on it. And this one has a point. This one over here looks kind of like a little needle or pin pad and so forth. This interferes with circulation because of the different sizes and shapes. This also interferes with oxygen transport. So it's called an anemia because these people, if they are in low altitude countries with high oxygen content, and if they don't exert themselves to a great extent, then they generally can live a period of time. But this blood condition causes wide-reaching effects on their overall health. So we look here. These are crystallized, taking odd shapes, clogging circulation, and leading to a variety of organ-based damages. So breakdown of red blood cells, physical weakness, anemia, pale color, feeling of lassitude, heart failure from the increasing heart action can all result from these breakdown of red blood cells. Also, blood cells break down enough, you can have impaired mental function. The clumping and the circulation, the clogging of the small blood vessels leads to heart problems. The undependable delivery of oxygen and nutrients can activate pain receptors, can activate the inflammation reaction and fever, can damage the brain as well as the heart, since these are two critically dependent. And the brain especially, mental function, paralysis, can be effects of the clumping. But also, we have a system where our normal red blood cells, they get old, they get broken down, they get frayed. A membrane will, turn up. And we normally want to detect those, filter them out, and replace them with new red blood cells. So this is what the spleen is doing, also the liver. But in the spleen, these sickle cells are so numerous that the spleen is overworked to get to a good proportion of normal red blood cells, and it can cause the spleen to be damaged. So sickle cell disease is something that, by the way, we identify this with black populations in America because of our history with slavery. We took slaves from areas that were high in sickle cell anemia, and that is true of all areas. It's not really racially based, it's based on the environment. So there are areas in sub-Saharan Africa, there are areas in the Middle East, and areas in India where malaria is very, very high. And the reason that sickle cell is present in high frequencies there is it turns out that this mutation, which produces a disadvantage in terms of oxygen transport, produces an advantage in terms of resistance to malaria. Malaria is a disease that depends on inhabiting the red blood cells and reproducing the malarial parasite. And by chance, this change that impairs your ability to carry oxygen is also a change that inhibits the life cycle of the malarial parasite. So wherever malaria is high and a significant mortality factor, we find high frequencies of sickle cell allele. Now, this is showing the effect of erythropoietin, EPO, something that we mentioned before. It is produced and released by the kidney. EPO goes through the blood to the bone marrow, where it causes an increase in the production of red blood cells and a restoration of hematocrit. Now, the reason this is shown this way, if you moved today from St. Louis, Missouri to Colorado Springs, 8,000 feet, you're living in the mountains. Pardon me. You're living in the mountains, and all of a sudden the oxygen tension is lower. You're going to feel, you go out for your normal walk and walk up a hill that you normally can handle fine. You're going to be panting because of the low hematocrit. Erythropoietin is what's going to adjust your red blood cell content and basically elevate it from your lowland hematocrit. Erythropoietin is something that's had some attention as a result of some medical treatments. Dialysis, the ability to keep a person alive when their kidneys are failing, was introduced, and the dialysis machine turns out differentially removes EPO. So it causes anemia in the 70s. This was an undesirable and unanticipated effect. Patients were given EPO, originally collected from victims of aplastic anemia. If they had a D12 deficiency, then their EPO is high. Or they had hookworm infections. That's destructive of red blood cells, and they're high. And this was continued until the AIDS pandemic, which now prohibits blood pooling. Recombinant DNA technology is something that has come to our rescue as we now produce this protein outside a living body, and it's given to dialysis patients, but also AIDS patients who become anemic, and those receiving some chemotherapies like Procrit. On the side of enhanced performance, it's also getting attention from athletes. It is an illegal application, but if you can imagine, if oxygen transport is limiting how fast you run the marathon, and you can temporarily increase your red blood cell content, it is a type of performance enhancement. Now, the red blood cell, as I said, numerous present everywhere, constantly turning over. There's no time that we say, well, let's not produce red blood cells. But we're always cycling them, starting up here in the upper right with this red blood cell production factory in the red bone marrow of all of our bones. So here come the new red blood cells. 90% will survive about 120 days, so 3 to 4 months. That, by the way, is the basis for the timing on the A1C. And about 10% normally break down as a result of circulation. Hemolysis is that process. Now, when a red blood cell breaks down, you're going to get fragments of membranes, and also you're going to get hemoglobin release. Hemoglobin and iron produce a toxicity problem, which is going to have to be dealt with. The 90% that are circulating are eventually going to be screened out and basically identified as damaged red blood cells by a macrophage, a type of white blood cell. The macrophage will use its lysosomes or its lytic enzymes to break down and release the heme group, and basically also take care of the solid debris. You can see the hemolysis, the damaged parts are going into macrophages as well. So the question is, where do we find a lot of macrophages? We find them in the lymph system, but we also find them in the liver and in the kidneys. So this processing of red blood cells releases heme. There is a significant, as you can imagine, reclaim of amino acids, since hemoglobin is a protein. When you break it down, a lot of amino acids are released. This is showing that those are reused. They would enter, they would not go straight to the bone marrow, but they would enter what would be called an amino acid pool wherever they occur. So any protein synthesis that's going on would draw on that pool. But this is a significant return of amino acids to the body. Now, heme can be broken down into iron. Here's the iron in ferrous form, transported in circulation by a carrier molecule called transferrin, where it is reintroduced into the manufacturing centers of the bone marrow in the formation of red blood cells. So here's the basic production. Now the waste side is a little more complicated because heme releases some protein fragments. Biliverdin and bilirubin, shown in green here, enter the general circulation from wherever we're breaking down the red blood cells. And they go to the liver. Bilirubin is excreted as part of the bile in the digestive tract. So that's how it enters the digestive tract and provides a coloring agent for the feces. That bilirubin that is not filtered out in the liver in the general circulation will be filtered out in the kidneys. You can see that some of this bilirubin is absorbed with the nutrients in the intestine, and it is filtered out in the kidneys, where it produces urochrome, the main yellow color of the urine, and where hemoglobin is also excreted. So here is the manufacture and breakdown cycle and the basically reclamation of waste products from the general circulation and their expulsion in both feces and in urine. So that completes a major portion of the blood chapter. I'm going to do a couple of things here. So I'm going to stop the recording.